The theme of my photographs this year is cell microscopy. I've always dreamt of taking a photo of a cell, but never thought it was possible. Every time I tried to think of a science photography topic, I kept on coming back to cells. So Mum encouraged me to email university laboratories to see if they would let me take photos of cells in the lab. I wasn't expecting any replies, so I was ecstatic when I heard back from labs at Melbourne and La Trobe universities. I feel so lucky to have taken micrographs with a light microscope and an electron microscope. Seeing light and electron micrographs in front of me inspired me to explore the differences between light and electron microscopy in detail. And that is the primary scientific principle in my photos. Some other scientific principles that I have touched on include cell structure, magnification, and refractive index. My photos show 2 micrographs from a light microscope and 2 from an electron microscope. I chose them because they are a good demonstration of the differences between images produced from light and electron microscopy. Some of the many differences between the 2 microscopy techniques are visible in my photos. Light microscopy generally have lower magnification and resolution than electron microscope. That is best seen comparing picture 2 and 4. Picture 2 is a fluorescent micrograph of a cell and picture 4 is an electron micrograph showing part of a cell. You can see in picture 2 that the mitochondria are visible but not in any detail. But in photo 4 you can see the mitochondria in great detail, even down to the double membrane and wave structure inside the organelle known as the cristae. The electron microscope takes pictures by detecting electrons that have been shot through the sample in the microscope. This results in the image being monochrome in contrast to light microscopes that use a light source to view a sample so micrographs can have color. This video shows how I took electron micrographs. I moved the Joy-Con to the center the cell, and then I turned the knobs at the left of the Joy-Con to increase the magnification. When I was happy with the image, I clicked Capture to take a photo. The process of taking a light micrograph was very similar. I would like to say a huge thank you to scientists Eric Hansen and Ted Johnson for inviting me to their labs and teaching me so much about cell microscopy. It has been a dream come true.